Uh, Today, Brian and I would like to take this opportunity to talk about our meeting in Santa Rosa that happened just a few weeks ago. This meeting was the annual California Big Data Workshop where all seven BDTA centers from California gathered to share our latest progress and discuss potential collaborations. Brian will start us off with a collaboration he proposed at the meeting, and I'll give a quick recap of the meeting and very briefly go over the other proposed collaborations.
Hi, great. So the collaboration I proposed was between our center, the Heartbeat UK Center, and the Cedar Center. And then after the proposal was made, Mobilize then suggested they'd also be interested in working with us on this project. And we were lucky enough to win the grand prize, which was actually a Mini Cooper, which is very exciting. So I'd like to show you the new car we got here. Yeah, so it's a little small, but it's still a Mini Cooper, so that's pretty cool. We won that, a great prize. Okay, so the basic idea behind the Cedar project, it's a web service where you can build metadata templates, and you can also populate metadata templates. So you have different templates describing the minimum amount of information to be released for a particular type of experiment. So our proposal was to use some of the natural language processing techniques that we're developing for Aztec for the extraction of metadata to be able to automatically extract data set metadata from publications to match the fields of these templates. So if somebody is writing a publication describing the release of a new data set, and they're going to populate a Cedar template to go along with it, we could potentially automatically identify the fields and extract some of that information from the publication to just help speed up the whole process of populating these templates. Let's see. Yeah. So yeah, as I said, with Aztec, we are getting a lot of the material for the Aztec database from publications in PubMed describing tools, and we're extracting all of the tool metadata from the PubMed articles. And we can do something very similar for to populate the Cedar templates. So as an example, the types of things we're extracting from publications, the tool name, the list of authors, institutions, tool description, the links associated with the tools, the funding sources, and the tags. And we're using GrowBid, which is a machine learning library designed specifically for publication mining in order to do this. So, yep, again, the general idea is to either get metadata either from articles that are on PubMed, or if there's a PDF that we could extract it from before the article is actually published, using our hybrid service to then populate the Cedar metadata templates. And this was the project outline that we presented at the Santa Rosa meeting. So our center, Heartbeat UK, would develop a REST API to extract and return dataset metadata from publications, again, either PDFs or directly from PubMed. And they would also provide us with user-entered data from their tool to help train our metadata extractor. And they would be able to call our new API from their metadata template tool. And then we could also help test the system by authoring metadata for cardiovascular proteomics datasets, which our center produces. So again, it's just a great way to help automate the system of filling out metadata templates, which might not be something that's possibly not that much fun. It could be a little bit of a routine, a little bit of drudgery. So hopefully we could automate this as much as possible and make it a smooth experience.
Okay, with that, does anyone have any questions about Brian's proposal?
I don't quite understand. So this is a proposal and it's planned, or this is approved and is going to start? I don't quite understand the setting.
So the proposal was so that there was sort of a competition at the Santa Rosa meeting where people would brainstorm different ideas for how different BUK centers could work together. And this is what we came up with Cedar. And then each person just presented on their proposal briefly at the meeting, and then they just voted on whichever ones they thought were the best possible collaborations. So this is what we outlined at that time. So it's still in the preliminary stage. So what we want. So during a year from now at the next California B2K meeting, we would sort of present on what we've done over the last year with regard to this collaboration. And we're also hoping during the all hands meeting to have it have it more developed by then, and be able to present at least a sort of a roadmap to this finished product at the all hands meeting. But it's still in like an early proposal stage.
And in the sort of final stage, or this will be is a proposal just for informal voluntary collaboration, or this is actually a funded activity then?
So yeah, to my knowledge, I don't think we're planning on submitting a request, a funding request or anything like that for this project. I think it would be funded just under the Cedar and HRP2K's existing grants. So this
I
is,
see.
yeah, this was.
This is structured collaboration, basically.
Yes, yes.
Okay. I understand. Is there
So this is
anything on expected volumes? Do you have estimates on how many, units, so let's say in the Aztec context, then resources you would expect to mine? Do you have, say, preliminary data on what is the potential target space or something like this?
No, and I mean that's a little bit difficult because so the Cedar project right now is not in a open access. It's still in like a private beta phase, so there's not really a lot of people populating these templates right now. So yeah, so we don't really have a lot of that information right now.
I see.
This is Andrew here. Just to confirm, the Cedar templates, those are reference biomedical ontologies. Is that correct?
Yes.
So, so they define the templates, and I thought what Cedar was going to do is, I don't know, build like things that make autocomplete easy and make finding the correct ontology. Is that also correct?
Yes. So
Okay.
the idea is that we could tie the autocomplete to the actual. So if they already have a draft of a publication that's going to accompany the release of this data set and the formal description of the data set, they could use that publication to populate these fields ahead of time.
Okay, and then the NLP you're planning on doing also then needs to be aware of the ontologies that were used to define the template. Is that correct?
Yes. Yes.
Okay. And then how does Mobilize fit into this?
So that's so they just expressed they have some people working on similar similar projects doing text mining, and they're just interested in joining this project with us. So they would probably be on the working with us on the NLP side of things, um, but that's not totally defined at this point.
Any other questions? Okay, then I'd like to give a quick recap of our California Big Data meeting in Santa Rosa. So this meeting was held just a few weeks ago, where all the BDK centers in California came together to share and promote their most recent work and have discussions on how we can leverage the California-based BDK efforts into further consortia activities for large-scale biomedical science and training. The meeting was a two-day event in which the first day consisted of presentations from each center about their work and then demos of all the new applications that have been developed. And then on the second day, we presented potential collaborations in hopes of winning and driving back with a brand new Mini Cooper. As you learned, Brian came home with the prize. And in total, there were 11 proposed collaborations. I won't go through all of them, but I'll just highlight two. Let's see. So one of the proposed collaborations I like to mention is the case study involving California BDTK centers. This was proposed by Dr. Jack Van Horn from the BDDS Center. The idea is that we have centers studying brain and cardiovascular diseases, and we want to be able to apply each center's expertise to tackle these problems using the resources we've created. Jack outlined some of the basic steps we could take, including storing the data and metadata using the BD bags developed by BDDS, and then creating new pipelines using the tools and from each center, as well as sharing workflow protocols, and then develop new metadata ontology standards that can be saved using the Cedar tool, and then validating each approach with new data. And the short-term goal for this project would be a possible position paper on the unique role that California BDTK centers have for such a research project, the special place that the California population has for such a project, and the novel contributions to national cardiac and brain health through such a project. And another proposed collaboration is linking.
Sorry, may I interrupt?
Yeah, go ahead.
Can you go back?
Mm-hmm.
So there's basically one word in this which is somewhat specific. The rest is a fairly generic outline of a collaboration. But unfortunately, I don't understand the one word which is specific, namely BD bags / mini ID. What's that?
So I think the BDDS Center has developed a system to identify data sets and kind of group data sets for a particular experiment into one what they call BD bag, so that they can those data sets can just easily be passed around and mentioned in publications or on web services.
Okay, thank you.
Yeah. Any other questions about this proposed collaboration? Okay, so the next one, another proposed collaboration is linking Panther with GeneWiki project proposed by Jessica Lee from our BDTK Center. So Panther stands for Protein Analysis Through Evolutionary Relationships, and it is a project from the BDS Center that serves as a classification system to classify proteins in order to facilitate high throughput analysis. And these proteins are classified according to their family and subfamily, molecular function, biological process, and pathway. And in this collaboration, an API would be developed to automatically populate gene ontology and annotation to GeneWiki. And the aim is to enhance the Go annotations on GeneWiki pages, as well as expand gene orthologs using the Panther phylogenetic tree relationships. Yeah, so if you'd like to see the rest of the collaborations or learn more about them, you can access these slides, which will be posted with the recording of this video on YouTube. Yeah, and with that, I'd be happy to take any questions. So thank you everyone for attending this call. This concludes today's conference.